keynote address, we are very lucky to have a very, very experienced scientist, a very experienced researcher, a very experienced academician in the name of Professor Yukio Yamori. Professor Yukio Yamori is a professor emeritus, Mukogawa Women's University in Japan. Professor Yamori is a pathologist, nutritionist, epidemiologist, and preventive medicine physician. There's an educator also, an institute administrator, and a consultant. Professor has received numerous awards in the course of his career. He has contributed to research on the pathogenesis of hypertension, stroke, and atherosclerosis. He's also contributed in gene analysis of cardiovascular diseases, development of models for cardiovascular disease and disease prevention, and cardiovascular and nutritional epidemiology. Professor Yamori will be discussing a WHO coordinated that's cardiovascular disease and alimentary comparison study, which covered over 60 populations in the world. This study revealed or gave evidence on the involvement of sodium intake and the sodium-potassium ratio. The sodium and potassium were analyzed based on 24-hour urinary samples, and these were associated, sodium was positively associated with age-adjusted mortality rates of stroke. The sodium and potassium ratio also had a direct correlation and was a predictor of the occurrence of strokes. Since the cardiac study also proved significant inverse relationship of the 24-hour urinary biomarkers for fish and soy intake, that is, fish and soy intake reduced the risk, including the intake of isoflavones and taurine with age-adjusted mortality rates of coronary heart diseases. The study investigated the association of these biomarkers with the risks of cardiovascular diseases. His focus this afternoon will be to contextualize, put this study in context within the health gaining domains.
Mr. Chairman, ladies and gentlemen, it's my great pleasure to talk about cardiovascular risk factors with a focus on 24-hour urinary biomarkers of fish and soy intakes as useful predictors in the context of healthy aging. Firstly, I'd like to congratulate the first nutrition and dietitian conference in Kenya is organized successfully by Kenya Nutritionists and Dietetics Institute and Journal of Nutrition and Dietetics. Our worldwide study covering over 60 populations in the world was successfully carried out also in Tanzania and Nigeria in Africa by the great cooperation of our many friends in the world. This study in the world showed daily traditional dietary customs were important for the longevity in Georgia, living in Georgia, the former name, and Uyghur people in Silk Road, area, as well as Okinawans living in Okinawa, and the Okinawans living in Hawaii, Okinawans immigrants living in Hawaii, who reached the longest average life expectancy in the late 1980s among the Japanese in the world. The diets related to Japanese longevity in the center are sushi, fish on vinegared ice, and soy products, tofu mixed with vegetables, a typical Okinawan dish. Japanese diets were recently listed as UNESCO's intangible cultural heritage. Japanese are well known for their longevity. The average life expectancy for both sexes has been #1 in the world. Particularly, Japanese females have maintained #1 position for over the last 2 decades. Are diets and nutrition related to Japanese longevity? Yes, we can answer now definitely from our studies in the world. Our worldwide epidemiological studies were started from our successful development of rat models for research on hypertension and stroke. Stroke-prone hypertension rats, which we established in 1973, genetically developed hemorrhagic and ischemic stroke without exception, and contributed worldwide not only to the exploitation of new antihypertension agents, but also to gene analysis for the prediction and nutritional prevention. And nutrition such as adverse effect of salt and beneficial effect of fish, magnesium and potassium index were proven experimentally in these animal models. The model was called stroke-prone spontaneous hypertension rats, shortly SHRSP. They quickly developed severe hypertension and stroke in 100% when fed on low protein, high salt diet. But when they were fed on high protein, high salt diet, the incidence of stroke reduced down to 10%. When they were fed on low protein, low salt diet, the incidence of stroke was 80%, and high protein, low salt feeding prevented stroke. Such extensive experimental studies proved nutrition could prevent stroke. We reported our experimental studies on the nutrition prevention of stroke at WHO Expert Committee on Cardiovascular Disease in 1982, when the dietary goal of daily salt intake was decided for the first time as 6 grams a day. However, no data were available worldwide how much salt was taken actually by different populations in the world. Therefore, we developed special device to collect 2.5% of the voided urine each time, as shown here, just by pushing this button buns on the urine in the small tube inside drops into the under compartment, and the rest of the urine is discarded. By repeating these simple procedures at each time of voiding urine for 24 hours, we can exactly estimate our daily intake of salt, vegetables, fish, soy, and so on by analyzing their biomarkers such as sodium, potassium, taurine, isoflavones, and magnesium. The WHO cardiac study is the acronym of the WHO coordinated cardiovascular disease and alimentary comparison study, which I proposed to WHO in 1983 for analyzing the biomarkers of the diets in association with blood pressure or cardiovascular disease mortalities in the world. Thanks to the supporting donation to WHO from 300,000 companies and industries, we could successfully carry out health examination in 61 populations of 25 countries, which were classified into 3 groups. Populations marked with large green circle are regions with food culture for healthy longevity. Green dots encircled with red regions with deteriorating food culture, and the large red circled regions with food culture unfavorable for longevity. About 100 males and 100 females aged 48 to 56 were randomly selected from each study site for blood pressure measurement, 24-hour urine collection, blood tests, and questionnaires, and over 14,000 participants were examined for over the last 20 years. We used standardized automated blood pressure measurement system to avoid observer bias. Our first health survey for Maasai in 1987 demonstrated their blood pressure was very low and they had no high blood pressure. In contrast, Tibetans living at the foot of Mount Everest had the highest prevalence of hypertension. We asked them to collect whole-day urine. Maasai used no salt at all at the time, and Maasai's daily salt intake, mainly from milk, was very low, only 2.5 grams, the lowest in the world. Tibetans took 16 grams of salt in average a day, and many suffered from severe hypertension over 200 millimeters of mercury. As shown here, the mortality rate of stroke, mostly caused by high blood pressure, was significantly positively related with 24-hour urinary salt excretion. This data indicate reduction of salt intake down to 5 grams a day, as recommended by WHO, should greatly contribute to the prevention of stroke. Stroke mortality, as well as bedridden disability and cerebrovascular dementia, for which stroke is a major cause, are expected to be greatly decreased if daily salt intake is reduced down to less than 7 or 8 grams a day, as recommended by Japanese government. Stroke is one of the major cardiovascular diseases, and coronary heart disease is another major cardiovascular disease. This study indicated coronary heart disease mortality was to serum cholesterol levels. Japanese cholesterol levels are low because their main source of energy is rice containing no cholesterol. Since women are protected from hypercholesterolemia in pre-menopausal period, coronary heart disease mortality rates are clearly lower in females, around 30 to 60 or 40% of those in males in all cardiac study populations. Clear gender differences in coronary heart disease mortalities are noted either in Scotland or Japan, where the mortality rate is highest or the lowest in the world, respectively. We also noted menopausal rise in blood pressure was significant in Western countries, where the soybean containing isoflavones with weak estrogenic activity were not eaten. In contrast, blood pressure rise at menopause was not significant in Asian countries, in China and Japan, where the daily soybean consumption was high and urinary isoflavone excretion biomarkers of the soy intake was higher than 10 micromole a day. Then we investigated soy isoflavones, the structure of which are similar to the estrogen, female sex hormone, and isoflavones have a weak estrogen-like activity, so that soy isoflavones may be protective against coronary heart diseases. Our cardiac study demonstrated first time in the world that urinary isoflavone excretion was inversely related to the age-adjusted coronary heart disease mortality. The data indicate about 20 micromole isoflavones excretion in the urine, corresponding 40 milligrams of isoflavones contained in 60 to 75 grams of dry soybeans is associated with low coronary heart disease mortality in the Japanese. We further analyzed the mechanism, how isoflavones work on the cultured endothelial cells obtained from the aorta. We could demonstrate isoflavones like estrogen increase the activity of nitrogen monoxide synthase, NOS, to produce more nitric oxide to dilate blood vessels and to inhibit thrombosis at endothelial surface. Since nitric oxide is destroyed by oxygen radicals, the effect of soy isoflavones should be increased by the combination of the antioxidant intake from vegetables and fruits. I would like to explain the mechanism of isoflavones effect by using cartoon by a famous Japanese cartoonist, Sanpei Sato. So soy isoflavones taken from the diets are absorbed into the bloodstream and act like female sex hormones to dilate blood vessels and inhibit blood clotting by producing nitrogen monoxide. Since nitrogen monoxide is destroyed quickly by oxygen, antioxidants such as vitamin A, C, and E are needed to maintain the function of nitrogen monoxide. Therefore, the intake of soy plus vegetables and foods improve bloodstream to the skin, as well as the heart, the kidney, and the brain. We may conclude that soybeans, which keep skin pretty, activate vital organs and contribute to longevity through the improvement of peripheral blood circulation. Our cardiac study further demonstrated a significant inverse relationship between urinary isoflavone excretion and the age-adjusted mortality rates of prostate cancer, high in Western countries, but very low in Asian countries. Isoflavones with weak estrogenic activity have the beneficial effect on the suppression of the prostate cancer cells. Similar inverse relationship was observed between urinary isoflavone excretion and the age-adjusted mortality rates of breast cancer. The mortality rate was the lowest in Japanese and Chinese, where a lot of soy products were consumed daily, and urinary isoflavone excretion was higher in Japanese and Chinese women so far examined in the world. The beneficial effect of isoflavones on the breast cancer can be explained by the estrogen receptor occupancy with isoflavones, which block the strong effect of estrogen causing breast cancer. Experimentally, we could observe the addition of isoflavones to the culture medium of human breast cancer cells blocked the growth of the cancer cells caused by strong estrogen, as well as by endocrine disrupting chemicals which activated estrogen receptors. Moreover, our cardiac data indicate the mortality rates of cancers of all sizes was significantly inversely related with 24 urinary isoflavone excretion. This inverse relationship can be explained because isoflavones are proved to inhibit angiogenesis, new blood vessel formation caused by cancer cells, and to attenuate the growth of cancers by the blood supply reduction. As for the health of the elderly people, bone fracture caused by osteoporosis is the second common cause of the bedridden disability following stroke in Japan. Our study in Japanese elderly over 70 living in Hawaii showed the participants with high bone density excreted more urinary isoflavones than those with lower bone density, indicating enough soy isoflavones intake, like estrogen, might reduce calcium resorption from the bone. Therefore, soy intake is recommended to prevent osteoporosis or extending healthy life expectancy. Therefore, we should eat soybeans to become healthier, but only 6% of soybeans produced in the world are eaten by humans. Soybeans are used as feeds for cattle, and humans who are eating too much meat from cattle are often suffering from obesity and lifestyle-related diseases. Since cattle eat 8 times of soy consumed by one person, just for producing meat for only one person, more soybeans should be eaten directly by humans for supporting protein to 7 billion people living on the earth who should share together the limited amount of protein. The beneficial effect of isoflavones on the breast, in contrast to soy-eating Oriental population, we noted high risks of lifestyle-related diseases in non-soy-eating populations, such as Australian Aboriginal living in Melbourne, who were proved to have the highest risks of cardiovascular diseases, such as extreme obesity. And our cardiac study showed low magnesium intake was related to a higher risk of obesity in Aboriginals, and over 1/3 of Aboriginals had high blood pressure at the age of 20s. These high risks of Aboriginals are ascribed to the rapid transition of their bush food to westernized food stuff such as flour, salt, sugar, oil, and fat given as welfare support. Their traditional bush food, such as caterpillars, seashells, kangaroo meat, nuts, and berries, are apparently rich source of variety of nutrients, particularly magnesium. Since our WHO study clearly demonstrated 24-hour urinary magnesium was associated with coronary heart disease mortality, 24-hour urinary magnesium excretions were divided into 5 quintiles in the world. The results indicate in the lowest first quintile, the percentage of the obesity, hypertension, and hypercholesterolemia were significantly higher than in the fifth quintiles, as shown in red, yellow, and orange columns. In contrast to urban Aboriginals, rural Aboriginals living near the sea were engaged in the culture of ales and had relatively lower cardiovascular risks. They were eating seafood containing taurine. Taurine is a simple sulfur amino acid rich in fish, shells, squid, and octopus, and has a beneficial effect on blood pressure and serum cholesterol level, and therefore atherosclerosis, which were all proven experimentally in our rat models. The risk of dyslipidemia was clearly lower in the Aboriginal, whose 24-hour urinary taurine to creatinine ratio was high, indicating fish eating custom is good for health in Aboriginals. Since our WHO study clearly demonstrated BMI, systolic and diastolic blood pressure, and total cholesterol levels were significantly higher in the people whose 24-hour urinary taurine excretion were lower than the average of the taurine excretion in the world. Less fish intake was related to obesity, hypertension, and hypercholesterolemia. Obesity, hypertension, and hypercholesterolemia are the major risk factors of coronary heart diseases. Therefore, the age-adjusted mortality rates of coronary heart diseases was significantly inversely related with 24-hour urinary taurine excretion, indicating the more the seafoods were eaten, the less the mortality rates of coronary heart diseases. The seafood intake of the Japanese were highest and followed by Mediterranean populations. The consumption of fish 80 to 100 grams a day appears to be associated with low coronary heart disease mortality rates in the Japanese. "24-hour urinary taurine excretion of the total participants in cardiac study were divided into 5 quintiles. In comparison with the highest 5th quintiles, the lowest first quintiles showed higher odds ratios in obesity, hypertension, and hypercholesterolemia, as shown by red, yellow, and orange curves, respectively. These results indicate both 24-hour urinary taurine and magnesium reduced cardiovascular disease risks. Therefore, the highest quintiles of 24-hour urinary taurine and magnesium were compared with the lowest quintiles of 24-hour urinary taurine and magnesium. The odds ratio of obesity, hypertension, and hypercholesterolemia were 6.3, 1.8, and 4.6 higher in the lowest first quintile than in the highest 5th quintile, confirming the benefits of taurine and magnesium intake for cardiovascular disease risk reduction. We may understand the importance of both magnesium and taurine from the prehistoric evolution of life and nutrition of human beings. Bush and kitchen-maiden foods contain rich magnesium and taurine. Since life was first born in the sea 3.8 billion years ago as a single cell, and finally as a process of evolution of our ancestors became hunters and gatherers, food gatherers. We human beings used to take enough magnesium and taurine, which supported the physiological function efficiently inside the body of our ancestors, probably gifted with thrifty genes, which enabled them to survive with their limited amount of food. Our worldwide data indicate the evolutionary concept of human nutrition should be reconsidered for developing healthier lifestyle without metabolic syndrome. This is a food pyramid of the Japanese diet. Japanese longevity depends on the lowest coronary heart disease mortality rates among developed countries in the world. Cardiac studies suggested the importance of cardiovascular protective nutrients from soybeans and seafood, the biomarkers of which were magnesium and taurine. These prehistoric nutrients for humans containing much magnesium and taurine are still daily available in Japanese, and the merit of this Japanese food are rice, soy, seafood, and seaweeds rich in magnesium and taurine. However, the definite demerits are excess salt intake and calcium deficiency due to the lack of the customs to drink milk and to eat daily dietary products. These demerits increase stroke and osteoporosis, two major causes of bedridden disability, and therefore shorten the Japanese healthy life expectancy. Since Japanese are customarily eating soy containing magnesium, we notice significant positive association between urinary soy isoflavones and magnesium. Our worldwide 24-hour urinary analysis revealed the obvious characteristics of Japanese diet. Biomarkers of the soy and fish intakes, isoflavone creatinine ratios, and taurine creatinine ratios were divided into 5 groups, each from small to large quantity intakes, and the all participants were divided into 5 by 5, in total 25 groups. This graph indicates the ratio of the Japanese in each one of 25 groups. The highest soy and fish taking group contains nearly 90% Japanese, indicating Japanese are commonly taking both soy and fish. The lowest intake of taurine and magnesium intake taking group, there was no Japanese. Higher soy and fish intakes are related to the lower coronary heart disease mortality. As our WHO cardiac study first demonstrated, coronary heart disease mortality rates were inversely related with the biomarkers of seafood and soy, taurine, and isoflavones. The average life expectancy is inversely related with coronary heart disease mortality rates, and the lowest coronary heart disease mortality in the Japanese was related with the highest life expectancy in Japanese. Therefore, fish and soy are important in the Japanese longevity. However, these traditional foods are becoming to be consumed less among the younger Japanese. Among Japanese, Okinawans living in Okinawa, the islands located in the southern Japan, are well known for their longevity. Okinawans women were enjoying the longest life expectancy in Japan and in the world in the early 1990s. Their traditional daily food consists of lots of soy products, local vegetables rich in antioxidants, seaweeds, and fish. Despite the longest life expectancy of female Okinawans in the world, Japanese immigrants from Okinawa living in Brazil have been strongly influenced by Western dietary customs, as shown here. They got accustomed to eat a lot of beef called churrasco with excessive rock salt on it. Japanese immigrants' cardiovascular disease risk factors such as hypertension, hypercholesterolemia, and obesity in Hawaii and Brazil were definitely increased than those in Japanese living in Okinawa. Particularly in Japanese, lifespan became shorter by 17 years than that of Okinawans. Moreover, the tendency to diabetes shown by higher glycohemoglobin levels, hemoglobin A1C, was markedly increased in Japanese immigrants living in Brazil. 20 to 25% of the people showed the high blood sugar level in Brazil. Actually, we noted traditional foods such as soy and fish were consumed less among Japanese immigrants living in Hawaii and Brazil. Okinawans in Okinawa were eating soy and fish enough because their 24-hour urinary excretion excreted isoflavones and taurine. However, Japanese immigrants, particularly in Brazil, consumed less soy and fish as their low urinary isoflavones and taurine indicated, and their average life expectancy became 17 years shorter than the longest Japanese life expectancy in the world. We therefore tried to make bread containing soy and fish nutrients in cooperation with local bakeries for reducing the risk of high-risk populations such as Scottish people, Japanese immigrants in Brazil, and Australian Aboriginals. For example, we carried out 8-week intervention study in Japanese immigrants living in Brazil by offering them soy bread containing 25 grams of soy protein a day, and the bread containing 2 grams of fish oil, DHA, a day. And we proved both bread decreased systolic blood pressure significantly in 8 weeks. We also confirmed non-HDL to HDL ratio, that is atherogenic index, indicating the proneness to develop atherosclerosis decreased about 10% from the baseline level in both soy and DHA bread intervention groups. This 10% reduction of atherogenic index correspond to the gender difference in the index, high in males and low in females. Since atherogenic index may be related to the aging of blood vessels, the improvement of atherogenic index corresponding to the gender difference between females and males is expected to the prolongation of life expectancy 6 or 7 years or longer. Since soy and fish nutrients improve the risk of high-risk populations, we also conducted an intervention study on the effect of low-salt, vegetable-rich, healthy lunch boxes in 70 Japanese businessmen for 1 month, and compared soy plus fish lunch boxes with meat lunch boxes in 2 groups. Both soy plus fish lunch and meat lunch reduced body mass index and diastolic blood pressure significantly from the baseline because of the reduction of caloric intakes and salt intakes in both lunch groups. Moreover, soy plus fish lunch reduced atherogenic index significantly, nearly 10% more than the meat lunch from the baseline. This result again indicate possible prolongation of life expectancy by taking soy and fish daily. And if such lunch, even once a day, is continued for lifelong, 7-year prolongation of life expectancy may be expected. For starting soy intervention study for high-risk Australian Aboriginals living in Melbourne, we explained soy would be good for health and longevity in Aboriginals because soy contains enough magnesium, fibers, and flavonoids like their traditional nuts and berries. And after health examination, 33 volunteers were randomized into 2 groups, given for 8 weeks either 25 grams of soy protein containing bread or placebo wheat bread. The results of this intervention study showed significant reduction of diastolic blood pressure, indicating isoflavones in soy protein bread might increase nitrogen monoxide production to reduce blood pressure. Atherogenic index, the ratio of the bad LDL to good HDL, was significantly decreased in soy bread groups, indicating isoflavones may increase LDL receptors of the liver to increase LDL incorporation into the liver and to reduce LDL level in the blood. We finally started a population approach to cardiovascular risk reduction in 2002 in Hyogo Prefecture, located at the center of Japan, the population of which was 5.6 million, corresponding 1/20 of all Japanese populations. This population approach, called Healthy Hyogo 21, emphasized the dietary goal to eat rice with soy and traditional Japanese food such as fish and vegetables, as well as to reduce salt intake based on the results obtained from a worldwide epidemiological study. When we started this, we selected 3 urban towns, 3 farming and 3 fishing villages to monitor the effect of the population approach by health surveys and 24-hour urinary analysis in about 1,000 people each year. This data shows that the distribution of 24-hour salt excretion during the initial 3 years of the campaign in 2002 and 2004. The distribution of salt intake shifted toward left, and the people taking salt less than 10 grams a day gradually increased. The average daily salt intake was significantly reduced from 12 grams to 10.1 grams. The percentage of the people taking less salt than 10 grams a day was initially only 1/3, but increased over half in 2 years. Correspondingly, both systolic and diastolic blood pressure of the people monitored for the first 3 years was significantly decreased, and the percentage of the borderline hypertensives with systolic blood pressure were from 140 to 160, initially 23% was reduced significantly down to 17%, indicate 1/4 of the borderline hypertensives became normotensives by the 3-year campaign. Since over 10 years passed after the start of the population approach, we collected 24 urinary samples as a health survey from the people aged 30 to 69 in order to estimate the effect of 10-year campaign for eating well-balanced, low-salt rice, soy, and traditional Japanese food. The data of women aged 40 to 69 showed salt intake in average was significantly decreased, and soy intake was increased 1.7 times higher than the baseline levels. Concomitantly, we noted significant reduction of both systolic and diastolic blood pressure for the past over 10 years. Moreover, triglycerides were decreased significantly, and HDL cholesterol levels, that is good cholesterol, were significantly elevated, indicating the phytoestrogenic effect of isoflavones. We also noted significant reduction of fasting blood sugar. The reduction means the improvement of insulin sensitivity. Fish intakes were divided into 3 groups according to 24 urinary taurine to creatine ratios. Highest fish consumers, the third quartile, showed significantly higher HDL cholesterol, good for preventing atherosclerosis and coronary heart diseases. Soy intakes were also divided into 3 quartiles of isoflavones in 24-hour urine, and the third quartile showed significantly lower insulin resistance, which corresponded to the lower fasting glucose levels in the blood. This indicates higher soy consumption is good for the prevention of diabetes. Traditional fish and soy intakes are good for the prevention of atherosclerosis and diabetes. However, high fish consumers showed significantly higher salt intake, 1.7 gram more than the first quartile, and high soy consumers showed significantly higher salt intake, 1.9 gram more than the first quartile. Since high salt intake increased hypertension and stroke, traditional fish and soy diets should be taken with optimal salt intake. Otherwise, the risk of stroke is increased in the Japanese. As for salt intake, we Japanese, enjoying the longest average life expectancy in the world, should learn from Maasai people living at the border of Kenya and Tanzania. Their salt intakes in average in the right was the lowest. Returning back to the merit of the Japanese diet, we further studied the effect of concomitant intakes of soy and fish. Soy and fish intakes were divided into 3 groups: low, middle, and high intakes, according to the 24-hour urinary isoflavones and the taurine excretions. In the group taking highest amount of both soy and fish, HDL cholesterol levels were significantly higher than in the group taking the lowest quantity, indicating the concomitant intakes of soy and fish showed increased good HDL cholesterol to prevent coronary heart diseases. This dietary increase in HDL cholesterol corresponds to the gender difference in HDL, which may affect the males and the female difference in the life expectancy. Since Japanese women's life expectancy is 7 years longer than that of the males, diets may be hopefully extend life expectancy 7 years at their best. We further studied also the effect of concomitant intakes of soy and fish. Soy and fish intakes were divided into 3 groups: small, moderate, and a large quantity according to 24 urinary isoflavones and taurine excretion. In the group consuming the large quantity of both soy and fish, folic acid in the blood was proved to be the highest. Folic acid is a kind of vitamin known to be important in the development of nervous system and in reducing the coronary heart diseases. Folic acid is recently expected to prevent dementia in the elderly. Therefore, both soy and fish intakes are recommended to extend healthy life expectancy. This study also confirmed both fish and taurine and soy isoflavones in 24-hour urine were associated significantly with magnesium excretion, indicating fish and soy intakes attenuate cardiovascular risk through increased magnesium intake. Therefore, cardiovascular risk reduction associated with fish and soy intakes may partly be affected by increased magnesium intake and concomitantly increased intakes of magnesium-containing vegetables and dietary fibers. Urinary biomarkers of sodium and potassium are well known to be related to salt and vegetable intakes, affecting the risk of hypertension and stroke. In addition to the balance between sodium and potassium, the present worldwide study indicated urinary isoflavones and taurine were closely associated to the dietary consumptions taking fish and soy, and thus affected significantly coronary heart diseases and atherosclerosis-related diseases, as well as some types of cancers. Magnesium intakes, which are related with soy, fish, and other beneficial foods such as seaweeds, and dairy products, should also be a urinary biomarkers for cardiovascular disease risks. Therefore, 24-hour urinary analysis are regarded as useful for evaluating individually the actual nutritional situation related to cardiovascular disease risks, and the analysis on urine samples are expected to contribute to the prevention of cardiovascular diseases. Finally, I will show the usefulness of urine analysis for evaluating population's nutrition in the context of longevity. We carried out a health survey for the Japanese immigrants over the age of 70 in Hilo, Hawaii, who reached the longest average life expectancy in the world among the Japanese in 1980s. We compared them with the Japanese living in Amino, Kyoto, Japan in 1995, and the numbers of the participants in the parentheses and their average age nearly 77 to 78 are shown here. The result of salt intakes in Hawaii and Kyoto are plotted on the data showing the association of salt with stroke mortality. Since salt intake in Hawaii was lower than Okinawa and Kyoto and close to the goal of salt intake of WHO at that time, 6 grams a day, the mortality of stroke should be lower in Hawaii, and this was the positive data supporting the longevity of the Japanese immigrants in Hawaii. The reason why Japanese in Hawaii naturally could reduce their salt intake lower down to near the goal of WHO is, I suppose, the influence of Polynesian cooking like lau, steamed meat, steamed vegetable meat in Hawaiian cuisines, as shown here. By steaming food, we can easily reduce salt intake because most vegetables are eaten so tasty without adding salt during cooking. We revisited Japanese immigrants in Hawaii last month in April. As shown here, we noted they were still keeping their good dietary custom to enjoy a lot of fruit salad, wild and sea vegetables, steamed food, soy products, and raw fish mixed with sesame and vegetables. Soy and fish intakes are related to the lower coronary heart disease mortality. As our WHO study first demonstrated, coronary heart disease mortality rates were inversely related with soy isoflavones and seafood taurine. The data of the Hawaii and Kyoto, Japan were plotted on the worldwide data. These data confirm that soy intakes in Hawaii was higher than in Kyoto, Japan, and fish intake in Hawaii was somewhat lower than in Kyoto, but still higher than in Western countries. The data of Hawaii was related to their lower coronary heart disease mortality, which might contribute to their longevity. Our comparison between Hawaii and Kyoto, Japan revealed two important longevity-related dietary factors. The first was lower salt intake, proved by 24-hour urinary salt analysis, that is 6 grams a day, in the elderly of Hawaii. The second was higher serum protein levels in Hawaii, where people were taking soy, fish, pork, meat, and also milk products. These two dietary factors may contribute to the higher cognitive function in elderly in Hawaii. Our long-lasting study in Kyoto, Japan indicated serum albumin, the component of serum protein, was significantly positively related with cognitive function of the elderly 7 years after the albumin was checked by our health examination. Higher albumin may contribute to the better cognitive function. Another long-lasting epidemiological study of the Japanese elderly in Fukuoka, Japan, reported the people taking more Japanese foods such as soy, fish, vegetables, and seaweed, plus milk products, were suffering from nearly 50% less dementia than the people who are taking less soy, fish, vegetables, and milk, indicating proper mixture of the Japanese traditional diets with Western diets is ideal for healthy longevity. In conclusion, Japanese in Hawaii are expected to be long-lived because of healthy lifestyle, especially diets, less salt, more vegetables, and fruits, which can be detected as lower sodium to potassium ratios when urine is checked. Moreover, their diets contain more protein and people are physically active and less smoking, better than Okinawans in Okinawa, Japan. Finally, so far we noted from our health survey nearly 20 years ago, people in Hawaii were keeping strong family and community relationships such as Okinawans and Japanese associations, which are now evaluated as social capital important for healthy longevity. All these factors seem to contribute to better cognitive function and less dementia in the elderly in Hawaii. This message from Hawaii is important not only for healthy longevity in the Japanese people, but also for the people in the world. where the elderly populations is now rapidly increasing. In conclusion, cardiovascular health and aging must depend on the metabolic syndromes such as obesity, hypertension, dyslipidemia, and diabetes caused by insulin resistance. These risks increase stroke and coronary heart disease through the acceleration of atherosclerosis and disturb healthy longevity. As food factors for longevity and cardiovascular health, soy containing isoflavones and protein, and seafood fish containing taurine and DHA are important. Japanese life expectancy, average life expectancy in males and females has been the top in the world, and Japanese traditional dietary customs for eating soy and seafood, not only fish but also seaweeds, together with rice and vegetables daily, except for excess salt intake, are apparently appropriate for reducing the risk of metabolic syndrome. Although the sodium should be reduced worldwide, vegetables, fruits, nuts, and beans containing potassium dehydration should be increased to reduce the adverse effect of salt. Low salted intakes of soy, seafood, fish, and seaweeds, as well as vegetables, which can be checked by urinary biomarkers as the predictors of cardiometabolic risks, will hopefully control these metabolic syndromes and finally contribute to healthy longevity. I would like to close my lecture by emphasizing Japanese diet relation soy and fish contribute to the healthy longevity. Traditionally, fish eating culture has been important for us Japanese. For example, the decoration of sardine was used to be hung at the entrance of each home on the New Year day of old lunar calendar in the beginning of February, because people believed the sardine protects us against demons entering into the house. Indeed, modern nutritional science indicates DHA and taurine, rich in fish, protected us from coronary heart diseases as a major cause of death in the developed countries. Soy eating culture has also been regarded as being good for health and fortune in Japan. We used to celebrate New Year days of our old lunar calendar by eating as many soybeans as our own age plus one in the hope of healthy longevity, and also celebrate New Year days by throwing handfuls of soybeans, shouting, "In with fortune, out with demons." 2 handfuls of soybeans are nearly equal to 70 grams of soybeans, containing 25 grams of soy protein, which is recommended as a daily soy intake for preventing atherosclerosis and coronary heart diseases by the FDA of USA. I hope 2 handfuls of soybeans taken daily from either traditional soy products or new soy food will help us to call our fortune of long life in by driving out the demons of lifestyle-related diseases now prevailing all over the world. Indeed, raw salted soy and fish keep the doctors away. Thank you very much.
Why the Japanese have a very long life expectancy. If you recall, he said the life expectancy for the women is about how many years? 87. Ours in Kenya is how long?
Thank you very much indeed. Before applauding this lecture, just I'd like to introduce this animal cup, which was used to estimate dietary intake. Exactly, precisely. Maasai people use this, enjoy using this, and we found their salt intake was very low. So in the future, for the improvement of dietary question, you have a big nutritional program. So that I do hope nutritionists in Kenya will utilize this scientific evaluation of dietary intakes. Then you will contribute to the improvement of nutritional situation in Kenya to attain the longest life expectancy in the world. Thank you very much.
So, for purposes of those who may have missed out on the reading, we don't think we have much to give Professor, but we have given him a desk pen, a plaque, which is for government. It has a coat of arms, and we have KNDI logo. We want KNDI to be on his desk in Japan. And simply cardiovascular risk factors with a focus on 24-hour urinary biomarker at fish, the way it is. And this is in the first conference of nutrition and dietetics, the inaugural conference for nutrition and dietetics science. We are very happy to present this, and Elena and Professor would come